Am I audible and visible? Let me know, then we'll start. Okay, fine. Uh, Hello everyone, welcome to my class. I'm myself Dr. Deepthi Karya. I'm the PhD Physiology and your educator for Physiology. Uh, Today we'll discuss about sarcomere, one of the short note may be asked in your exams. Okay. Now, here in this channel, I'm uploading videos for first year MBBS nursing, physiotherapy, and homeopathy students. And if you wish to complete your whole syllabus of physiology, if you are supposing MBBS or nursing or physiotherapy, homeopathy stream, you can join the channel and you can complete your course within a given period of time before your exams. Okay, so please subscribe the channel for the same and watch all the videos. Fine. Now, if you wish to get soft copies of all my notes, that is available on the application, that is Vesalius. This application you have to download from the Play Store and you have to subscribe. Second thing, if you wish to get notes, hard copy of the notes, that is also available, and that is in the form of physiology notes. Fine. That is available on Flipkart as well as Amazon. Link for all that is given in the description box. Now let us start with today's topic, that is sarcomere. Okay, now sarcomere, yesterday we have discussed, those who have watched yesterday's videos, they come to know that we have discussed skeletal muscles. Skeletal muscles, they are striated muscles. You can see striations are there. Okay, and striations are because of this A band and I band. Skeletal muscles, they have actin and myosin filaments, and their arrangement that gives striated appearance. Fine. Now, what is sarcomere? Now we will discuss sarcomere. That is the distance between two Z disc or Z line. Okay, so here you can see this is the sarcomere. It is also known as structural and functional unit of the skeletal muscle. Okay, now, so here presence of, in this sarcomere we have presence of, you can see dark and light band. This is light band, this one, which contains only actin filament. Okay, but if it contains myosin filament also, that is your dark band. Okay, dark band is also known as A band because it is anisotropic to polarized light. Length of the A band, that is 1.5 micrometer. Okay, and in the center of A band, this one you can see here, this is H zone, which is also known as Hansen's zone. This one, Hansen's zone. Okay, this is less refractile region. Why is it less refractile? Because here head part of the myosin filaments are not present. Okay, only this tail part, and actin filaments are also not present in this Hansen zone. Okay, and in the middle of H zone, this one, you can see here, there is M line. Okay, M line due to central bulging of the A band. Okay, and this M line, it becomes more prominent when your muscle contracts. Okay, another is pseudo H zone. Pseudo H zone means this M line. Hello, Lingaraj. Okay, hello beta, welcome to my class. So here you can see this M line. What is pseudo H zone? Pseudo H zone, okay, is equal to M line plus narrow light area on either side of the M line. So in the center of H, there is pseudo H zone. Fine. Okay. Now, so this was about dark band, we discussed this one. Okay. Now I band. I band, why is it known as I band? Because it is isotropic to polarized light. Isotropic to polarized light. Okay, length is 1 micrometer. Okay, A band length we have discussed, that is 1.5 micrometer. Okay, and in the center of I band, this is total I band, this one, in the center we have Z disc or Z line. So you can say that Z line divides I band in two equal halves. Okay, fine. Now next, as we have discussed, sarcomere, that is the functional and structural unit of the skeletal muscle. It is the part between two Z disc. Fine. Now we discuss what are the different lengths, how lengths of sarcomere changes. Okay, you can see here, this is the sarcomere as we have discussed, this one between two Z line. Okay, now at rest, as we all know that this is A band and this is I band. So what we discussed, that was at rest, A band length, that is 1.5 micrometer. Okay, and I band length, that is 1 micrometer. So total length of the sarcomere at rest is equal to 2.5 micrometer. Okay, and during muscle contraction, as we have discussed, this actin filaments, they overlap the myosin filaments. You can see here. So this length decreases. Okay, this length, how much it decreases? Complete actin filaments, they overlap the myosin. So it may be, you can see here, actin filament or A band may be absent like this with complete contraction. So maybe as low as 1.5 micrometer. Fine. Okay, but the length of A band will not change. A band length remains same. Okay, fine. Now during stretching, when the muscle is stretched completely, you can see here like this highest. So what happens, there is no overlapping between these two. Okay, so it may be as high as 3.5 micrometer. Okay, this is the length of skeletal muscle or sarcomere of the skeletal muscle when it is completely relaxed. Fine. Now we'll discuss about various filaments of the sarcomere. Okay, these filaments, they form contractile apparatus of the skeletal muscle. Fine. These are made up of contractile proteins. Contractile proteins are our actin and myosin. Fine. You can see here, this is actin filament. What are the components of actin filament? I will discuss. Actin filament. Okay, and this is your myosin filament. Myosin filament is made up of molecules. Okay, molecules, they unite to form one filament. Filament is, hello Pragati, filament is made up of molecule. You can see here multiple molecules, they form a filament. So this is actin filament, this is your myosin filament. So contractile protein is actin and myosin. We will come to meromyosin also, don't worry. Then regulating proteins or regulatory proteins, these are your troponin and tropomyosin. We will discuss. This is your tropomyosin and troponin, how they regulate, that also we will discuss. And anchoring protein. Anchoring means they provide strength. Which are the anchoring protein? All the three are important: contractile proteins, regulatory proteins, and anchoring protein. Try to remember the names of the anchoring proteins. Actinine. This is actinine. Okay, Pragati, welcome to my class. You are physiotherapy student. I think these classes will be very much helpful to you. Actinine, then titine. We will discuss all this. Nebulin and dystrophin. Okay, this all are also anchoring protein. We will discuss one by one all the proteins. Fine. Now, so you can see here, this is actin. Okay, troponin and tropomyosin, we will discuss in detail. Now, I'll just show you. Now, so first we discuss about thick filament. As I told you, thick filament consists of, these are your thick filaments, this all. Thick filaments, they are made up of your contractile proteins. Okay, myosin mainly. Okay, myosin, they are made up of myosin. Hundreds of molecules of myosin are there in the myosin thick filament. Okay, diameter of the thick filament is double than thin filament. Thin is your actin filament. Fine, and it is responsible for, it forms A band here. Okay, now If you take a cut section, you can see here, this is myosin filament. This myosin filament is surrounded by actin filaments. You can see on all the 6 sides, everywhere. You can see here, this is only this part. But if we are taking section of this part where A band and I band, they are in contact, you can see here actin filaments, they surround myosin filament on all the 6 sides. Okay, so you can see A band is surrounded by 6 actin filaments. If we take a cut section, I'm talking about cut section, fine. Okay, and if we are talking about one thick filament, this is myosin filament, it contains about 500 myosin molecules. Okay, now we discuss about the structure of myosin molecule. You can see this is one molecule. You can see here multiple molecules. These are molecules. They unite to form a filament. This one. Fine. So first of all, we'll discuss myosin molecules, how they work. Myosin molecules are made up of the myosin molecules. They are 10 to 11 nanometer thick and 45 nanometer apart. Okay, their molecular weight, if you can remember, 4,80,000. It has got six polypeptide chains, two heavy chains, and it has got two light chains. Okay, this two heavy chains and four light chains, two on each side. Okay, so 6 polypeptide chains. Heavy chain molecular weight is 2 lakh. Okay, so 2 lakhs plus 2 lakhs is equal to 4 lakh. And light chain molecular weight is 20,000. 20,000 multiplied by 4 is equal to 80,000. 80,000 and 4 lakhs. So the total molecular weight is 4,80,000. Fine. Okay. And your heavy chain, this heavy chains, they wrap around each other and they form double helix. You can see here, this is double helix. Okay, they form the tail part of this myosin filament. Head part, that is formed by your light chains. Okay, globular head is formed by the light chains. You can see here this side. Okay, fine. Now position of myosin molecules. This myosin molecules, you can see here, they are arranged in such a way that this head part, it points outside, okay, in one direction along one half of the filament. Okay, and they form cross bridges. You can see here, cross bridge means when the head hinges with the body part and tail part. Okay, this is cross bridge. It has got two hinges. One, you can see head here, okay, and one that is from the body. Okay, there are 2 points where each cross bridge is flexible and they are known as hinge between arm and the body and arm and the head. You can see this is head, this is arm, and this is body. So between head and arm and between body and arm, there are 2 hinges. Okay, now so you can see these are hinged arm. And what is the role of this hinged arms? They allow the head to be extended or just to move it or flex. Okay, so movement is possible. Okay, and globular heads, these globular heads, they project from the surface of the thick filament, you can see, towards the thin filament, and they bridge the gap between them. Okay, during when the muscle contracts, what happens, these heads, they are attached to the thin filament, you can see here, and what they do is they just move these thin filaments. Okay, so in thick filaments, you can say that myosin molecules, they aggregate with their heads in one direction, half of them, and other direction, opposite direction, other half. Fine. And what is their action? Because of this hinge joint, they just first bind with the actin filament and they move this actin filament medially. Fine. Now here now Pragati, your myromyosin has come. Okay, so when the myosin head is, or when the myosin molecule that is treated with the proteolytic enzyme, okay, commonly trypsin is used. You can see here this one. So when the trypsin is used, this is your myosin molecule. Okay, what happens? You can see here HMM and LMM, they are divided. HMM means heavy myromyosin and light myromyosin. Fine. So this light myromyosin, that is tail part. It doesn't have ATPase activity because ATPase activity is present in the head here. Fine. And heavy myromyosin molecules, they contain this globular head with ATP ATPase enzyme. Okay. Now this heavy myromyosin again, that is again split by the papain. You can see here this one. So it is again divided into two parts, that is fibrous and globular. Fibrous heavy myromyosin, that is your S2, and globular heavy myromyosin is S1. This globular has got, you can see, ATPase activity, but this fibrous doesn't have any ATPase activity. Fine. So this is all about your myosin molecule. Now we'll discuss about thin filaments, or you can say that actin filaments. Now this thin filament, they are made up of not only actin but troponin and tropomyosin. They form I band in the sarcomere, isotropic to polarized light. Here you can see this actin filaments, they are arranged, these filaments are arranged to form two chains of the globular unit. Okay, these are the globular units. Okay, fine. And their chains, helical chains are there, double helix is formed. Okay, and each chain, this thin filament, this one, your thin filament, this actin filament, each filament has got about 300 to 400 actin molecules. Okay, and if we are talking about tropomyosin filament, each filament contains about 40 to 60 tropomyosin molecule. Fine. Okay, now we discuss about actin. You can see here, this is actin filament. It has got diameter of 4 to 5 nanometer. This is all you have to remember. It is a double helix. Okay, it forms a double helix. Okay, and it extends from the Z line. Okay, it consists of globular actin, G actin molecules. Their diameter of one molecule, this one G actin, that is 5.5 nanometer. Their molecular weight is 60,000, and this molecule binds with the myosin head. Try to remember this thing. This is very important. If you want to know the muscle contraction, this thing you require to know. Okay, so it binds with the myosin head. This part, what? The globular part of the actin. Fine. Now, filamentous actin means this, all the globules, they form a filament. Okay, a fibrous actin. They are formed by polymerization. Polymer, multiple G actin molecules, they are polymerized. Along with that, when they get polymerized, there is energy liberation. Okay, so you can say that G actin ATP, that forms F actin plus ADP plus phosphate plus energy. So whenever your this F actin molecule is formed, F actin filament is formed, there is generation of energy. Okay, now so this is about your actin filament. Now next one, that is tropomyosin. This is, you can see this one, this red color one is, don't get confused. If your basics are clear, that becomes very easy for you to understand the muscle contraction and all. This is tropomyosin. This tropomyosin also is a filament which is made up of 40 to 60 tropomyosin molecule. This one has, okay, their molecular weight is 70,000. Okay, these are also filamentous structure, and they are present in the groove between the chains of actin. This you can see. So this is the actin, and on the actin we have tropomyosin filament. And this what is the important role of tropomyosin? They bind with the active site of the actin filament. So when this is here, this active site of the actin filaments are covered, so this actin cannot bind with the myosin. Therefore tropomyosin and troponin are known as regulatory proteins. They prevent binding of actin with the myosin. Otherwise what happens? Continuously actin and myosin, they got bounded, and that results in continuous use of energy. When it is not required, then at that time this tropomyosin prevents the binding. Another important protein, that is troponin. These are also small globular units. You can see here troponin. They are of three types: troponin T, troponin I, and troponin C. Okay, here you can see these are located at the interval along the tropomyosin here. So troponin T, this one, this troponin T binds to tropomyosin. Okay, please remember these are very important concepts. Tropomyosin, fine. Troponin C binds with calcium and troponin I binds with the actin filaments. Okay, and one important thing here, one molecule of troponin C binds with 4 molecules of calcium ion. Fine. Now, so what happens in resting condition? In resting condition, your troponin and tropomyosin, troponin binds with tropomyosin and this complex covers this active site. Okay, and this binds with troponin I binds with the actin. Now what happens when muscle contracts? This calcium ions, they bind with the troponin C, and when calcium ions they bind with the troponin C, what happens? This troponin tropomyosin complexes are removed from this actin molecule. So now your actin molecule, active site, they get open up, and therefore that results in binding of the actin with the myosin molecule head. Fine. So this is all about your regulatory protein. So this is the reason why they are known as regulatory protein. They will not allow the binding of actin with the myosin head. Fine. Now next, as we have discussed, 3 types of proteins are there. Number 1, that is your contractile proteins. Which are contractile proteins? Contractile proteins are your actin and myosin we discussed. Second, they are your regulatory proteins. Regulatory proteins are troponin and tropomyosin. Fine. And third variety are anchoring protein, means they strengthen your sarcomere. Okay, and one they are titin, nebulin, actinin. Okay, we will discuss. So these are starting with titin. This other name of the titin, that is connectin or gap filament. You can see here, this is the titin molecule. These are fine. Now these molecules, they are large elastic filaments and they connect the Z line to the, they interconnect the Z line, or they connect Z line to the M line here. Okay, what is their action? This titin molecules, they keep the myosin and actin filaments in their own place, and the side by side relationship between the myosin and actin filament, if this titin molecules are not there, that is difficult to maintain this actin and myosin filament to keep in the space, keep in their place. Okay, and this is achieved by this titin molecule. So if titin molecules are not there, you can see this actin and myosin molecules, they are not in there on their particular place. Fine. Molecular weight of titin molecule, that is large molecular weight, 3 million. Okay, and therefore it is the largest protein molecule in our body. Titin molecules are largest protein molecules. Okay, now again you can see here, these are the titin molecules. This one you can see here, this is okay. So these are like springy filament. The springy titin molecules, they act as a, what is their action? They act as framework. Okay, and by making the framework, they hold myosin and actin filaments in their own place. Okay, and therefore their contractile mechanism is also managed. Okay, one end of the molecule is attached to the Z line and other end, as I told you, it is to the M line. Okay, okay. Titin molecule itself also appears to act as template for initial formation of portion of the contractile filaments of the sarcomere. Especially they form a portion of the myosin filament also. So some of their action, that also it helps in the muscle contraction, also contractile process. Fine. But mainly they are your anchoring proteins. Fine. So that is titin. Second one, that is alpha actinin. This you can see. This is actin. This is not actin. Actin is a contractile protein. Actinin, that is regulatory. That is your anchoring protein. Fine. So actinin, it acts to anchor the thin filament. Okay, to the Z line. It allows the thin filament to bind the Z line, and it cross-link the actin filaments in the areas of the Z line. Then next is nebulin. I'll just show you. uh, these are your nebulin, this one. Okay, this nebulins, they run parallel and in close association with thin filaments, this one you can see here, in association with thin filaments, and they stabilize the length of the thin filament when muscle uh, develops and when muscle contracts. Okay, and this nebulin, they are inextensible filament, and this nebulin is connected at one end, you can see, to the actinin to actinin in the areas of Z line, you can see here at the Z line to the actinin. Okay, and sorry, at the other end they are attached with the tropomyosin troponin complex of thin filament. So nebulin is attached to Z line one side and other way to the troponin tropomyosin complex. Fine. So we have discussed anchoring proteins. One, that is titine, second, that is alpha actinin, third is nebulin. Fine. Next is dystrophin, one of the very important anchoring protein. Okay, this dystrophin I'll show you, uh, and the disease because of deficiency of dystrophin, that is also very important, Duchenne dystrophy. Okay, you can see here, this is dystrophin, this one. So dystrophin is connected to the thin filament one side, and to the other side it is connected to the this beta dystroglycan, this one. This is beta dystroglycan, this one. Fine, we will discuss. So dystrophin, that is rod like protein, large rod like protein, and that is anchoring protein that anchors the thin filament. So one side it is attached to the thin filament, and to the other side it is attached to the beta dystroglycan, and this beta dystroglycan connected to the extracellular matrix protein here, extracellular matrix protein, this is laminin. Okay, through alpha dystroglycan. Okay, so whatever force is given on this thin filament, this force is transferred outside. This is one of the very important anchoring protein. I repeat, dystrophin is a rod-like protein. It is connected to one side to the actin filament, and other side beta dystrophin, and this beta dystroglycan, and this beta dystroglycan is attached through alpha dystroglycan to the laminin. So it transfers the force from contractile system to the extracellular region through the transmembrane protein, and therefore it maintains the survival of the cell. And if there is defect in the dystroglycan or dystrophin, the disorder, as I told you, that is Duchenne muscle dystrophy, and that is characterized by muscle weakness. Fine. Now, other proteins are, we will discuss in detail about this dystrophin and this Duchenne dystrophy along with the disorders of muscle. Okay, other anchoring protein, that is desmin. I'll show you here. This is desmin, this one. This is okay. So desmin binds Z line to the sarcolemma. This is Z line, and on this here we have sarcolemma. So Z line connected to sarcolemma, that is by desmin. Then next is tropomodulin. This tropomodulin, that is located at the end of thin filament. This is actin filament. Okay, thin filament end. Okay, towards the center of the sarcomere, that is located. Okay, another one, that is myomachine and C protein. It is not properly visible. I'll just show you. Where is it? This one is myomachine. Okay, myomachine and C protein, that is also found in the thick filament here, myomachine. Okay, and here from M line binds to titine molecule and connect adjacent thick filaments to one another. So this is your titine molecule, you can see here, this one. So this myomachine, okay, it binds to the titine molecules and they connect adjacent thick filaments to one another. Fine. So these are about your different anchoring proteins. I repeat all the proteins. One, they are contractile proteins in the muscle. We have discussed they are actin, myosin. Okay. Second are regulatory proteins. Regulatory proteins are your can anyone? Regulatory proteins are troponin and tropomyosin. Okay, and anchoring proteins we have discussed, they are titin, okay, nebulin, alpha actinin, desmin, tropomodulin, and myomachine and C protein. Okay, so this is all about different proteins, and this is all about sarcomere. So if you have a question, you can write down this way. And again, the announcement is same. If you wish to get soft copies of my notes, that is available on the application Vesalius. You have to download the application and you have to subscribe the same. And if you wish to get hard copy of my all the notes which I am teaching in the lecture, that is available, physiology notes, that is a book. This book is available on the Flipkart and Amazon, and link for all this, our application, our book, that is given in the description box. As well as we also have practical application, Practicable, link for the same is also given in the description box. Thank you so much. Thanks for watching me. If you like my video, you can like it, you can share with all your friends and subscribe the channel. एंड हिट द बेल आइकन हिट क्यों करना है, बिकॉज मैडम जब भी लाइव हो रहे हैं तो आपको भी नोटिफिकेशन आ जाएगा मैडम लाइव है. जस्ट इट्स टाइम टू वॉच द वीडियो, इट्स टाइम टू स्टडी. ओके, थैंक यू सो मच.